Good day everyone, I am Maria Angela Denise Fajardo and I am going to discuss about Chapter 3, Lesson 3, The Roles of Genes in Heredity. Now, let us discuss what is heredity. Heredity is the passing on of traits from parents to their offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parents. Through heredity, variations between individuals can accumulate and cause species to evolve by natural selection. Now let us proceed to the roles of genes in heredity. The heredity information is contained or within the genes, located in the chromosomes of each cell. An inherited trait can be determined by one or many genes, and a single gene can influence more than one trait. A human cell contains many thousands of different genes in the nucleus. Heredity factors known as genes are thought to exist as discrete portions of chromosomes. The term discrete refers to the concept that genes are always located at the same point or on a chromosome. It is believed that pairs of homologous chromosomes contain linear, matching arrangements of genes exerting parallel control over the same trait are known as alleles, which are of two kinds, the dominant and recessive. Genetic characteristics can be extremely complex and may require the actions of several separate genes to be expressed in the adult organism. An example of traits that require more than a single pair of genes to be expressed are human height and human color. Other traits may be controlled by a single pair of genes. Some of these traits may have extreme phenotypes such as albinism or lack of skin pigment in humans. The following are the common genetic disorders. The first one is Down syndrome. The second one is the sickle cell disease. The third one is the phenylketonuria or PKU. And lastly is the hemophilia. Each of our gene codes contains unique protein, and each protein has a separate and distinct function to perform in the cell. Now let us proceed to the effects of genes in our environment. Genes are inherited, but their expression can be modified by interactions with the environment. Genetic traits are determined largely through the precise information found in the cell's gene structure. A variety of factors in the environment can actually alter the effects of a particular gene. Example of this is the effect of light or chlorophyll production. Although most plants have the genetic ability to produce chlorophyll, they will do this only in the presence of light. Without light, these plants produce only a light yellow pigment and therefore appear pale and sickly until they are exposed to sunlight. After a few days of exposure to sunlight, the chlorophyll production mechanism is enabled and green color returns. Cells store and use coded information. The genetic information stored in DNA is used to direct the synthesis of the thousands of proteins that each cell requires. The cell contains many thousands of such codes in its chromosomes. Each strand of DNA in the chromosome has the potential to provide the complete chemical code for the manufacture of at least one complete protein. These proteins are highly specific and they result in the expression of some specific trait or portions of a trait in the living and consequently in the organism of which they are part. Now let us proceed to gene mutations. Genes are segments of DNA molecules. Any alteration of the DNA sequence is a mutation. Usually, an individual cell's altered gene will be passed on to every cell that develops from it. Gene mutations may be defined as being any changes in the nitrogenous base sequence of a molecule DNA. When the base sequence of DNA is altered, the amino acid sequence of the polypeptide for which it codes will likewise be altered. Such an alteration may affect the operation of the resulting enzyme, preventing it from properly catalyzing its reaction and thus preventing a trait from being expressed by the cell. The majority of gene mutations are harmful because they result in the cell being impaired from performing some specific tasks. In rare cases, a mutation may result in a lethal gene that kills the cell either by producing a substance toxic to the cell or by failing to produce a protein of vital importance to the cell. Gene mutations are passed to every cell that arises from the mutated. If the mutation occurs in the somatic or body tissues, its defect is limited to the tissues immediately surrounding the mutated cell. If the mutation occurs in a primary sex cell, it passes on to the offspring that result from fertilization of or by gametes produced from the primary sex cell. A mutation can enter the gene pool of a population and be passed on to succeeding generations. This is known to be a cause of variation in a species. On the other hand, DNA is a polymer made up of a repeating chemical unit known as nucleotide. Thousands of the units are known to comprise a single DNA molecule, making it one of the largest of all organic compounds. DNA exists in hundreds of thousands, if not millions, of different forms depending on the precise arrangement of nucleotides in the molecule. Its variability is the key to genetic variation in living things. DNA nucleotides themselves are quite complex, being composed of three separate subunits. The first one is the phosphate group, a chemical group made up of phosphorus and oxygen. The second one is the deoxyribose, a 5-carbon sugar made up of carbon, oxygen, and hydrogen. And lastly is the nitrogenous base, a chemical unit composed of carbon, oxygen, hydrogen, and nitrogen. Bases found in DNA are adenine with the symbol A, thymine with the symbol T, cytosine with the symbol C, and guanine with the symbol G.
DNA code. The chemical and the structural properties of DNA are the basis for how the genetic information that underlies heredity is both encoded in genes and replicated by means of template. As we recall, the DNA is a complex organic molecules composed of thousands of repeating nucleotide molecules, and that each pre-nucleotide carries with it one of our nitrogenous bases. The particular sequence of introgenous genus bases adenine, thymine, cytosine, and genuine comprise a standard of DNA and providing the type of chemical code that is understood by the chemical mechanism of the cell. The DNA code is used by this mechanism to manufacture specific enzymes and other protein through the process of the protein synthesis. A DNA strand provides template for the formation of messenger RNA. The DNA code is transcribed RNA as the letter is synthesized in a pattern complementary to the DNA standard. The process by which the DNA code is transcription and molecules uracils nucleotides are substituted for DNA thymine. Each group of three nitrogenous bases, known as the triplet codon, provides the information necessary to code for the insertion of a single specific amino acid into building protein molecule. The particular sequence of triplet codons on DNA enables amino acid to be linked together in a specific sequence during protein synthesis. In conclusion, DNA is not merely a molecule with a pattern, it is a code, a language in a information storage. The genetic code. Every organism requires a set of code instructions for specifying age traits for offspring to resemble parents' information. Their parents' information must be reliably transferred from one generation to the next. Heredity is passage of these instructions from one generation to another. Discoveries in genetic science, desoxyribonucleic acid was revealed to be the chemically active agent of the gene. DNA replicates itself when chromosomes replicate in the early stage of cell division. DNA is passed from generation to generation during reproduction and acts as genetic factors. DNA interacts with the cell's chemical factory and produces the observable effect of the phenotype when genes are inherited by a cell or an organism. DNA regulates the production of enzymes in the cells and TRB enables the cells to perform the complex cellular chemical reaction necessary to sustain life. Stem cell therapy focuses on the use of stem cells to treat or prevent a disease or condition. Stem cells are a special breed of cells that are capable of differentiating into more than one kind of tissue. Arthritis, arthritis, cancers, bone marrow failure. Genetic engineering is a technique of controlled manipulation of genes to change the genetic makeup of cells and more genes across species boundaries.